So I've been in this job for quite a while now, a number of years, and I don't think I've ever heard a series of panelists so succinctly and comprehensively describe both the ecosystem for diagnostics and healthcare as well as the importance of diagnostics. And so I'm really impressed and really glad that we brought these four speakers to you today. So we're going to shift gears just a little bit for the remainder of the program, and if there's time for questions at the end, we'd be happy to entertain them. And certainly if any of our panelists can stay a little bit, they may be available as well, although I do think we have the room only until the half hour. So I just wanted to briefly describe a hypothetical case study and then ask each of our panelists, starting with Dr. Hansen, we'll work down the table and come back to Dr. Kavaz to finish up, to just speak briefly to how they would encounter such a patient or the samples from the patient in their system and how they would think through this diagnostic process and get to a therapy decision. So the hypothetical case study is that of a patient who lives in a nursing home for patients on long-term ventilators, but is admitted to the hospital for fever and low blood pressure. And blood cultures are drawn, and the patient is found to have C. auris, which was previously described by Dr. Kavaz. So if we could, I think we can just stay at the table and use the mics here, and if I might just ask, we'll move this other mic down the table to try to pick up on our video. Dr. Hansen.
Yeah, so can you guys hear me? I'm not sure. Oh, this one. At this point, it's a little bit easier for the infectious diseases clinician because he just told me what the patient has. They have a bloodstream infection with Candida auris. But I think the more difficult diagnostic dilemma for us occurs when the patient hits the emergency room in the ambulance. And they have a fever, they have low blood pressure, and we have no idea what is causing it. It looks like an infection, but is it a bacteria? Is it a virus? Is it a fungi? And we have very limited capabilities beyond viruses to get that answer in the emergency department. So what would happen for this patient in the emergency department, because low blood pressure is a medical emergency and there are protocols at our hospital and nationally that we would probably give this patient empiric antibiotic therapy. We would take our best guess at what he has or she has and prescribe broad spectrum antibiotics that cover the whole gamut of possibilities. The problem in this case being the patient didn't have a bacterial infection, it was a fungal infection. And not knowing more about the patient, we may or may not have guessed right in the emergency department about giving early antifungal drug. We know the longer you delay antibiotics or antifungals from the time the patient hits the emergency department or the blood cultures are drawn, the worse the outcome. So for bloodstream infections and bacteria in our hospital, we give antibiotics within 4 hours of hitting the door and often don't have an answer by then. For fungi, we may or may not guess right. And in this case, I don't know that we would have started empiric antifungal therapy. But now that we hear from the microbiology laboratory that we have a candida in blood, and we've got an identification of the candida, which we'll probably talk more about how that was made, we can guess based on this species that it will likely be resistant to at least one class of antifungal drug, and we would take our best guess at the drug that should be most effective, and that's in the candida class. But there are some Candida auris strains that are resistant to all of our antifungals. So we're in a holding pattern here for 24 to 48 hours to get the antifungal susceptibility results back from the clinical lab. And we take our best guess, get them started on that therapy, figure out where the infection was coming from. Is it deeper seated? Is there a collection of pus that needs to be drained? Did they have an IV that needs to be removed? What else can we do to help the patient get better? And I should have mentioned in my day-to-day work, we also partner very closely with hospital infection control. And we know that Candida auris can spread through health centers on the hands potentially of healthcare providers, and this would be an infection control issue for us. We would put that patient in isolation in a single room, and all folks who enter the room would wear gloves and gowns so we wouldn't hopefully transmit this fungus to people next door on our hands while we're performing patient care. So from my point, we'd prescribe the antifungals, we'd call hospital infection control, we'd alert the clinical lab, they would have alerted us, and now we're in a holding pattern for the antifungal susceptibility results.
So I'll tell you a little bit about where diagnostic tests are today for identification and susceptibility testing for Candida auris. So for identification, it depends on the platform. Like the older manual test kits that have a limited number of biochemicals, these typically will misidentify the organism as another Candida, or it's been shown to misidentify as Rhodotorula. So they don't do very well. The newer biochemical kits, we have a platform called VITEC2, and the YST card has 46 different biochemical tests. So this one is able to differentiate Candida auris from similar species, like it used to be confused with one called Candida hemoloniae, which is also can be multiply resistant, but it's not seen to cause invasive infections. So in our most recent software, we're able to differentiate Candida auris on that product, and it's already available because that product is exempt from FDA approval. In the early 90s, FDA decided to make biochemical identification test kits exempt from FDA approval, exempt from 510K approval. The MALDI-TOF platform, since it's a new platform, FDA required that 510K be submitted for any new claims or any new applications. So we have Candida auris in the MALDI-TOF research database, but in the IVD database, it's been added to the next software version, which will be put through trials and then submitted to the FDA probably sometime later this year. FDA have relaxed the rules for 510K approval of MALDI-TOF because they consider MALDI-TOF to be the standard of care, the state of the art. So it's easier to get a faster approval on something like Candida auris nowadays. For susceptibility testing, unfortunately, most of the products, the commercial products, they have to have approval on each individual species that's been tested. A certain number of susceptible, certain number of resistant isolates. So Candida auris has not been tested on these products, and it's not approved for use. It can be tested as a different Candida, and you could get results, but that would be off-label use. Or you can use a conventional type of method like Kirby-Bauer disk diffusion or conventional broth microdilution tray in order to get results. And yes, Candida auris is typically resistant to triazoles and can be resistant to other antifungals as well. So it's a very serious antimicrobial resistant problem.
Thank you. So at the state level, our role is really twofold. There's the laboratory side, and there's the epidemiology side. As Kim mentioned, it's a hospital-acquired infection, and so that's something that we're always very aware of and play a very, very active role with hospitals to deal with. In New York, as of last week, we have 65 clinical cases, including one infant. And we're currently aware of 62 involved facilities in 7 counties, which include hospitals, long-term care facilities, nursing homes, and hospices, where C. auris patients who we know were infected have resided. So why is that important? This means that, we know that in 2 of these nursing homes where infected patients have been, there are 44 cases where patients are colonized. Not infected, but they don't have symptoms, as it were, but they're colonized. This means that we're probably just seeing the tip of the iceberg here. Now, I'm not sure why New York is winning the race on the number of C. auris cases, but I don't think it means that we're really bad. I think it means that we're looking very intensely. But it does mean that the problem is probably quite widespread. Where one of the New York cases that we have is resistant to all 3 drugs, which is clearly very worrying in terms of treatment. The problem is really exacerbated because the most vulnerable people that are infected are often quite elderly, and they have many underlying health conditions. which means they're in and out of hospital and nursing homes all the time, which raises the potential for transmission. So it's something that we have to be very careful of. In New York, many of the hospital labs and commercial labs don't have the capability to distinguish C. auris from other Candida species. So we're actually doing a lot of testing for hospitals at the moment. We're using both culture on very special agar. We're using a real-time PCR assay that we developed, and we're using Malditoff as well. We're using PCR really as a screening technique to just see the prevalence, how much of it is out there. We're actually working on a screening protocol, an admission screening protocol with a number of our hospitals downstate, where we will use PCR just as the patient is admitted to a hospital to see if they are colonized or infected. Because if they are, then we can really quickly say this patient has to be isolated and therefore cut down the transmission rate. This is something that we're piloting in New York in collaboration with CDC, and once we know how this is going to work, we'll be rolling out our PCR as a screening tool, and then we'll institute it at many other hospitals around the state. So what we're trying to do is be very practical, very pragmatic, limit the spread of infection, putting out guidance to hospitals to say, hey guys, you need to be aware of this. When you have a patient, you need to isolate them, you need to clean surfaces, you need to wear the right PPE, protective equipment, to stop the spread of the organism. So there's epidemiology working very closely with the laboratory to mitigate what is a really quite worrying situation.
Thank you. So in this instance, we at CDC would be consulting with the states. We would hear about it through the state health department, or we might get the call from Kim, in which case we would look in the state health department, and we would offer our technical assistance, basically ready to get on the phone and discuss the case, advise on the epidemiologic and laboratory investigation, on the need to conduct an environmental investigation, sample the healthcare settings where the patient had been, look for additional cases, and if the state or city would ask us on invitation only, we might deploy technical staff to help with that investigation. Might depend on whether they've had C. auris earlier or not, and the circumstances of what's going on there. The other thing we would do, so both on the investigation, tracking the infection, understanding where it was acquired, but also advising on effective control measures and trying to contain it. At the same time, we would request an attempt to get an isolate so that we can conduct the whole genome sequencing and understand whether it's related to what might be happening in the region or where it might have come from. Also to add the isolate to our isolate bank, the FDA CDC isolate bank, that, like I said, we make available then for investigation and study by partners, including private industry for development of tests or therapeutics.
I want to thank you all for responding to that hypothetical case study and talking about some additional perspectives on how to manage a patient in that kind of scenario, and talking about some of the broader implications as well. So we have a couple minutes left. We'd be happy to take some questions before we wrap up. I'd throw it open to the floor. Sure.
What work remains to be done in terms of stewardship and oversight of data and computational resources? For instance, databases or software from a grant that then loses funding or it was shuttered. And what are the priorities in terms of accessibility to those resources, say if they're commercialized or proprietary?
So from the New York perspective, if we're using public money, it's public data. And it's available. We certainly share with the CDC, we deposit all sequences in NCBI, public money, public data.
And I guess the data at the hospital level is a little more proprietary, where we keep track of all the invasive infections that we've seen in our healthcare system. We keep track of patterns of antibiotic susceptibility and resistance, and it's called an antibiogram. And we update that every year, and we do it in a way that, you know, what's in the emergency room might be different than what's in the cancer center, might be different than what's in the HIV clinic, so we have site-specific antibiograms where clinicians can go to say, okay, this is what my patient has, this is the most likely susceptibility profile, but that's unique to our health system and not typically shared outside of that.
Could I just add one thing to what I said? This is all de-identified. There's no link to the patient, so they're not privacy issues.
And I would just note that your reference to stewardship programs, of course, is critical, right? And that's a good practice that's continued to spread and be operationalized in healthcare systems, as not only awareness of these challenges becomes more acute, but people understand the kind of practices that really need to be taken systematically in healthcare systems to address these issues. Sure.
How widely available are these new diagnostic technologies to states and other countries currently?
I think there's not one answer to that, but I'm sure maybe some folks,
I
Dr. Buzz
think
might
it
characterize that.
varies by countries and by setting. It varies. Some are available, others not. I mentioned when we were in West Africa doing Ebola, how we took the portable sequencer there, was used. Increasingly, I think there's an effort to try to develop point-of-care tests that can be available around the world.
And there are a number of kind of near patient or point of care molecular diagnostic tests that are CLIA waived, meaning that it doesn't have to be a skilled laboratorian to perform the test. It could be a medical assistant in a clinic. And those technologies are coming down in price and more readily available than some of the more complex whole genome sequencing, aside from the field sequencer, still are more relegated to the reference lab setting in the current time, at least in this country.
I would just make a general comment that availability of diagnostic technologies varies a lot depending on how complex the technology is, what kind of setting it needs to be performed in, the skill of the operators and other people to use it. A huge push in diagnostics, of course, is to develop the point of care tests that you've heard talked about here today, which is to bring tests that not only can provide rapid results, but can be performed by people who don't have high technical training in performing diagnostic tests. But the other thing to remember, especially for healthcare systems in lesser developed countries, is it's not enough just to have the diagnostic technology, even to have a really great diagnostic technology. You still have to have other systems in place to bring those tests to patients, to run the test, to get the results back to people that can actually use them, to make the proper diagnosis and have access to the right treatment protocols and therapies and so forth, right? So there's a much larger set of complex challenges that diagnostics fits into, but those advances in diagnostics are enabling of a lot of, then again, really positive things that can be done to improve patient care. I'm afraid we've run out of time, but if then you have burning questions and you'd love to come up and talk to our panelists, I'm sure they'd be happy to try to stay around a little bit. Join me in thanking our panelists. Thank you.